Hello, welcome to single-cell multiome analysis series with SciDAP. In the previous video, we started the WNN cluster analysis and we did it in two different settings. So we started two samples in parallel. One had an equal contribution of ATAC-Seq part and RNA-Seq, and the second one had less contribution of ATAC. Let's compare them. So, as in any cluster analysis we had before, this one has the information about the QC metrics. The first thing we see that our sample with the dimensions set as 50 and 50 has more clusters. There are 11, 13, and 13, and in the second sample it's 9, 12, and 13. Then we could look at the QC metrics. Then we could look at the clusters themselves. They look slightly different, but both of the samples look good. The clusters are distinct, and they don't look like a single bulb of intermixed cells, and this is a good sign. This sample also has positive silhouette score on average, even though some clusters are on the negative side still. Majority of them are very distinct, have very high silhouette score, especially in the resolution 0.3, because there's less clusters. And the same for the second sample. I think this resolution does not look as great. Then we move on to the Split by Dataset tab. And there, again, clusters for every sample. Here we see that the clusters are different. We saw that already. Remember, we saw it in our RNA and ATAC part. This is the cluster 3 that looks different. And let's look here. This cluster is also present, but this time it's on the other side. We also see another one that is differential. I'm curious what cell type could that be. Here we have genes of interest, expression, and coverage, and the gene markers heat maps. There is gene markers table and the peak markers table. Now let's look at the UCSC browser. I select RNA to highlight expression of genes that I like. This is our WNN map. It automatically selects the lower resolution, so it's 0.3 resolution of WNN. We could change the color here. We could highlight the view by dataset. We could highlight by condition and see that the TRM group has this differential cluster. Here we could color them by RNA reads. So you could make sure that we have all the best quality cells, mitochondrial percentage, novelty score, ATAC fragments, peaks, TSS enrichment score, nucleosomal signal, all of these metrics. There are the similar information, but presented in a different way. Then you could color cells by the clustering method. So the WNN clustering, as I mentioned, incorporates both ATAC and RNA modalities. And the clustering would be different depending on how many dimensions we assign for each of these two parts. We could highlight the ATAC and RNA separately to see how they correlate with each other. We could even highlight cells by donor or batch. Then we could label them differently. For example, we could color the clusters as resolution WNN 0.3 Then label them as WNN 0.4, and you could see which clusters don't match. Additionally, you could change the view. For example, if you would like to see the UMAP for ATAC-Seq, you don't have to go back to your ATAC clustering analysis. Everything is already here. There are clusters for ATAC-Seq. Then one can color them accordingly so it looks nicer. And we also could see the cluster markers. Then, because it identifies the cell markers automatically, you have the list of them right here. It's very convenient to see how markers distributed between the clusters. Together with peak markers and genes of interest, it is helpful for cluster annotation. Remember we saved the loop file? You could download the loop browser from the official 10x Genomics website and open your sample there. It produces quite nice pictures. You could explore different features here. Sometimes it is still not enough for the perfect figure. Some clusters are needed merging, or you'd like to recalculate gene markers and update your RDS file before publication. For these last improvements, you could use the manual cell type annotation pipeline. I will review this pipeline in the next video. It will be our last in the series. Thank you for watching.